Hello, Bloodstream listeners. If you joined us last year, you heard us dive into the exciting details of Hemgenix approval for hemophilia B. Since then, innovation hasn't stopped or slowed down. Research continues to leap hurdles, and we're so excited to continue to explore what's next. In this segment, we're asking, where is the science heading? We know Hemgenix is here, but what comes after? On today's episode, we're going to sit down with a researcher and a genetic counselor to take a look at how gene therapy for hemophilia began and where it could be going from here. For decades, the bleeding disorders community has been at the forefront of research and innovations, and there have been many humans and four-legged friends who have helped make this possible. So, as we kick off this new year, we are so excited to dive into conversations with experts in gene therapy to explore what we've learned within the bleeding disorders community and how it can make an even bigger impact, both inside and outside of it. First, hello! My name is Hanna Beary, and I just celebrated my first year as the Patient Advocacy Manager here at Believe Limited. And let me tell you what, wow, has it been weird? Has it been emotional? And by golly, it has been just about the best time of my life so far in my 29 years of age. I grew up hearing the stories of my cousins passing away from contaminated blood products that was supposed to save their lives, a story that I'm sure many listeners can resonate with me today. Since graduating college in 2017, go Hawkeyes! I've worked boots on the ground for bleeding disorders organizations, helping with advocacy, camp youth programs, and various other needs, making sure always advocating for the health equity of the community and other rare disease groups. What I've learned from my time in bleeding disorders nonprofit work is that the work is not yet finished, and I am dedicated to continuing to educate myself and spread that information to listeners like you. Though, enough about me. Before we get back to discussing the ins and outs of gene therapy, this segment is brought to you by CSL Behring, a global biotherapeutics leader focused on serving the rare disease community by providing innovative therapies. They now have a first-of-its-kind treatment for hemophilia B. To learn more about this treatment option, visit beyondhemb.com and download the B Support app from the App Store or Google Play to stay up to date on relevant information and manage your treatment story. Once again, this segment has been brought to you by CSL Behring, which now has a first-of-its-kind hemophilia B treatment. Visit beyondhemb.com or download B Support wherever you get your apps for more information. To set the stage, let's take a moment to hear from Dr. Mark Kay, a professor of pediatrics and genetics, as well as the head of the Human Gene Therapy Department at Stanford University, about some of the incredible hurdles research has come within the bleeding disorders community.
Hemophilia became a popular disease to go after because, first of all, there was a large animal model of the disease that recapitulated the human disease well. There's a canine model. Secondly, we knew from genetics and from protein replacement therapies that we only needed to replace a relatively small amount of the coagulation factor to really alter the course of the disease. We could measure the protein product directly in the blood, which was the biomarker we needed to measure. We could measure coagulation, we could measure the clotting factor, and we knew what percent, how much clotting factor would result in how much improvement in the disease. One other point, we published papers showing that you could get therapeutic levels of coagulation factor 9 first in mice and then in hemophilia B dogs. And this was kind of the aha moment, because not only did we have therapeutic levels, it seemed to be sustained.
Fast forward to the early 2000s, scientists start the race on the first clinical trials. While they showed promises, challenges like immune responses to viral delivery systems initially slowed progress. But as we know, advancements in technology soon cleared those hurdles.
Unlike what was seen in animals, when we finally got to a therapeutic dose in humans, the factor 9 level started off to be therapeutic. It was around 10 or 12%. But then, unlike the animals, it actually fell and went back to baseline. And that was totally unexpected based on the animal studies.
By the mid 2010s, trials showed longer lasting increases in the factor IX level, giving patients the chance to move away from regular factor replacement therapy. Let's hear from Dr. Marquet about this.
And then there was a second vector that gained popularity called adenoviral vectors, and those could give therapeutic high levels of coagulation factor. There was some immunogenicity and toxicity, which also made it clinically irrelevant as the preclinical testing went. And then finally, there was a vector called recombinant adeno-associated viral vectors. Once people realized that it could be used safely to get therapeutic levels of clotting factor IX, first in mice, then in hemophilia B dogs, and then ultimately in humans.
In 2002, we hit a milestone when the FDA approved of Hemgenix, the first gene therapy approval for hemophilia B, a one-time treatment changing lives. Looking back at gene therapy's journey reminds us that the race is not over. It's a blueprint for what's possible as we keep jumping the hurdles of science and medicine. We are so excited to continue conversations today with Dr. Marquet alongside Kaylee Dollerschell, a genetics counselor at the Center for Cancer and Blood Disorders in Colorado, who can share more about the different cross applications of gene therapies.
In certain types of gene therapy, an individual may require going through something to prepare their body for gene therapy. There's certain types of gene therapy that are called in vivo therapies, where we are giving somebody their treatment directly into the body, and it hopefully goes to where it's supposed to and does its job. And there's not a lot of preparation that has to be done to the body to receive that treatment. Things like hemophilia A and hemophilia B are those kind of common examples in my world. There's also types of gene therapy called ex vivo, where we are typically needing to take your cells out to kind of prepare the cells with gene therapy outside the body. There's other technologies that are getting a little bit more creative, where maybe they're not specifically giving you back a full functioning copy of the gene that's impacted, but maybe they're just kind of going around the gene that's impacted altogether.
As gene therapy advances, researchers are exploring how these techniques could be applied to other conditions. Could approaches used in hemophilia help shape the future of other genetic disorders?
So, for example, there's a new treatment for sickle cell that is not necessarily giving you a genetic change or genetic copy of the sickle cell gene back in its full functioning copy, although some of them are doing that too. But this one's actually impacting another part of the DNA that increases your production of fetal hemoglobin, which ends up kind of helping your sickle cell disease in general. So it's just one of those that sometimes we're going directly to the source to try to give you a fix, and sometimes we're just kind of making a path around it.
There's a trial that I believe is going to start very soon, where the way they're going to do this is they're going to use a gene editing technology in addition to the gene therapy. So I'm sure that many of the listeners have heard of CRISPR or CRISPR engineering. And I just want to say there's all sorts of new genome editing tools that are being developed.
As gene therapy advances, so do the tools shaping the future, like CRISPR. Many have heard of it, but what exactly and how could it impact gene therapy?
With CRISPR, what people have done is they've engineered this Cas9 or caspase-9 protein to work in mammalian cells. And it will only cut DNA, or primarily cut DNA, when it's guided to a specific sequence in the genome. And basically then the Cas9 uses what's called these guide RNAs, or they're RNAs that actually will bind to the nuclease. One arm of it binds to the nuclease, and the other arm seeks out a sequence that it recognizes in the genome. And then it brings the nuclease to that site in the genome and can make a DNA break. And when the DNA tries to repair itself, because naturally our DNA gets broken, there's DNA repair mechanisms. If there's a lot of other DNA there, such as DNA coming in from the AAV vector, it will basically try to repair it and insert that. And I think that this is a space to watch, because there's many different ways to the expanding space. And I think something that it's very exciting.
Gene therapy isn't just the future, it's here transforming lives. With advancements like CRISPR and expanding applications, the possibilities keep growing. But science never stops, and neither will we. We'll keep bringing you expert insights, real stories, and the latest breakthroughs. Once again, I'm Hanna Beary. Thank you for listening to It's a Whole New World, a gene therapy segment. We'll see you next time.